Protein synthesis takes place on ribosomes. So now we're going to look at the components of the ribosome. There are complexes of RNA and protein, about 65% RNA, 35% protein. There is one large subunit and one small subunit. And the prokaryotic and eukaryotic subunits or ribosomes are here with mass with respect to comparison. Prokaryotes 70S, eukaryotics 80S. This is the mass in Daltons. And again, there are 2 subunits per ribosome. These are the components of the E. coli ribosome. So the 70S ribosome is the whole complex. The larger and smaller units exist independently unless or when they're brought together on the mRNA when protein synthesis occurs. And here you also see the components of the 50S. It is composed of 2 different pieces and sizes of RNA in addition to 31 proteins. The 30S subunit is composed of 1 16S RNA and an additional 21 proteins. So quite a few components. The next slide shows an electrophoresis of the ribosomal proteins. So the ribosome was denatured, the proteins separated or degraded, the proteins separated, and then run out on a SDase phage gel. And so the components are, in this experiment, separated on size and likely charge as well. So now we're looking at the 16S RNA. That was the single RNA in the smaller 30S subunit. It contains 1,542 nucleotides and is one continuous strand of RNA. But you can see that even though it's single-stranded, it has significantly more than half of the bases in the RNA are hybridized to complementary bases in the same molecule. And what this does is form stem loop structures. So here's a stem, here's a loop, and we'll see that there are some functions for these loops, at least when we see them in a different molecule. But again, this is the E. coli 16S RNA. This is a closer view, and that reveals the complementation within the stems and shows also that it's not 100%. Most of the mismatches are GU base pairs, and here's a couple of those GU base pairs here, which will still be a pyrimidine purine pair, of course, but unusual pairing. The next slide shows a model of the ribosomal structure. And the 50S ribosome has been characterized by X-ray crystallography. And based on that data, this is the model of what that structure looks like. There are various proteins around the periphery with the 23S and 5S ribosomal RNAs at the core. The site of peptide bond synthesis lies within a central cavity that contains only the 2 ribosomal RNAs and no protein. So this is that catalytic site in the RNA contained within that ribosome and is responsible for the progression of translation. So the purple shaded areas are proteins. We have both white and orange indicating the 23S ribosomal RNA, the 5S RNA. The red here is the peptidyl tRNA, and we'll talk more about that in a moment. And the green is the aminoacyl tRNA. And I mentioned that the 50S has been the 50S has been well characterized, and this is data from extensive X-ray studies that have been used to construct the model of the overall shape of that ribosomal assembly. And the 50S basically looks like a baseball glove. And you could say that the 30S perhaps resembles a ghost, and the 70S, or the complete ribosome, top view, side view, has sometimes been likened to a mother and a child. Now we're going to look at transfer RNAs, amino acid activation, and the initiation of protein synthesis. So transfer RNA, or tRNA, are carriers of the activated amino acids. There are 30 to 50 different tRNAs. However, there is at least one transfer RNA for each of the 20 amino acids, although for many of the amino acids there is more than one. These are small structures, only 70 to 80 nucleotides in length, and they do contain distinctive rare bases that we will look at. They also, of course, not only carry, but they serve as carriers which recognize the codons in the mRNA. This is a diagram of a tRNA, or transfer RNA. There are 3 stem loop structures in all tRNAs. We'll talk about each one of them in turn. There's a DHU loop. Then the DHU loop contains a modified base called dihydrouridine. We'll take a look at that structure in a moment. There's the anticodon loop. So the anticodon loop contains a sequence that is complementary to the codon and recognizes a specific amino acid codon on the mRNA. There's also what is called the T pseudouridine C loop, and this is involved, sorry, I kind of went all over that. This is involved in binding the ribosome, and at the 3' end of this structure, 5' end being here, there is the site of amino acid attachment, and that has a sequence of C, C, A. So here's a comparison of the uridine residue to the high dihydrouridine residue. And if you look here, you can see where we have a double bond in the base here. It has been reduced in the dihydrouridine residue. Okay, so this decreases the ability to hydrogen bond, and while this part of the base is planar, this now is not planar. It's no longer planar. The other derivative of uridine is the pseudouridine residue found in that pseudouridine C loop. And again, by comparison, you'll note that the point of attachment changes, where here we have the NC or glycosidic link between the base and the ribose. We have in the pseudouridine residue a CC point of attachment. This figure shows you what the mRNA looks like in its folded form, and it looks more like an inverted L, if you will, kind of L-shaped. Here's our anticodon loop, the DHU loop, the pseudo T-pseudo uridine loop, and then again that amino acid point of attachment. This, and we'll talk about this again in a minute, this is a post-transcriptional modification. And of course, these mRNAs are also not transcribed with these rare bases in them either. But we'll talk about that again in a moment as well. So this is a precursor of tRNA. So this is an RNA transcript that contains a number of tRNAs with extra RNA between them. So the synthesis of tRNA is going to involve an interesting multi-step process. And the initial step are transcript holding the sequences of 7 tRNAs. The initial tRNA, again, contains the 4 normal bases, so extensive post-transcriptional modifications must take place to convert these bases to the rare bases that are found in the mature tRNAs. The individual tRNAs are excised from the transcript. The extra or non-functional RNA is just released. Also, as I mentioned a moment ago, the CCA nucleotides at the 3' end are not part of the initial transcript, so these must also be added by a post-transcriptional mechanism that we won't be looking at. This is just the amino acid modification that is added post-transcriptionally for that amino acid point of attachment. And again, that there's that 3' hydroxyl. I think this slide got replicated once too many times. Sorry about that. Now we're going to look at the 4 stages of protein synthesis. The first step is the activation of amino acids. So amino acid substrates undergo activation by the formation of the covalent aminoacyl tRNA. It is important that the correct amino acid gets charged on to the appropriate tRNA. There is a specific mechanism that is very accurate for the initiation of protein synthesis. Generally, this occurs at the first AUG next to a Shine-Dalgarno sequence in the mRNA, and we'll take a look at that momentarily. Elongation is an energy-requiring process which occurs on and is catalyzed by ribosomes. The elongation process requires an mRNA template and various charged tRNAs, of course. Termination of protein synthesis occurs when the translation machinery, the ribosome, encounters a stop codon in the mRNA. This figure just represents the amino activation mechanism or reaction. The covalent attachment of an amino acid to a tRNA is again an energy requiring process. We've got ATP involved here, and so the energy for this process is provided by that ATP, which gets hydrolyzed, and 2 inorganic phosphates are released. Also AMP, and an aminoacyl AMP serves as an activated intermediate. So we've got an intermediate here prior to the tRNA coming in and sorry, I didn't point out the enzyme, amino ACLT tRNA synthetase, and here it again is charging that tRNA with that amino ACL. So here is the tRNA that is now carrying that amino acid to be incorporated into a growing peptide chain. This tRNA synthetase utilizes both the functional groups and shape of the amino acid to prevent the attachment of an incorrect amino acid. It recognizes the anticodon, acceptor stem, and sometimes other parts of its tRNA substrate. That's the enzyme, keep in mind. Okay. So this is the activated intermediate, and this is the charged tRNA. In prokaryotic organisms, N-formyl methionine is generally the first amino acid at the N-terminal or terminus of the protein. And we're going to take a look at the formation or the transformation, the mechanism behind converting methionine to N-F-met and eventually a tRNA here. So in most prokaryotic organisms, an AUG codon just downstream from the Shine-Dalgarno sequence is recognized by a special tRNA carrying that N-formyl methionine, or F-met. It turns out that this AUG at the 5' end of the coding sequence is the only AUG in a gene that is read as F-met. The internal AUGs are all recognized by tRNA that carries the normal methionine. And again, this slide depicts the biosynthesis of F-met complex with the tRNA. It involves an extra step. The methionine tRNA is formed by a specific methionine tRNA synthetase, which is right here. And in the second part of the reaction, a formyl group with 1 carbon is added to the alpha amino group. So here's where that occurs, of the methionine residue by transformylase enzyme. Okay. So here we have our modified methionine, which will appear as the first amino acid in a prokaryotic protein. So we've talked about and seen the involvement of N10-formyl tetrahydrofolate before. This is just showing you this so you actually can see what this molecule looks like. Here's the formyl group, and it is basically functioning as a carrier of activated 1 carbon fragments. And it is also derived from methionine and serine. Excuse me. So let's take a look at initiation of protein synthesis, which of course is called translation. The initiation of translation occurs downstream from a purine-rich Shine-Dalgarno sequence on the mRNA. We've already talked about this sequence. This sequence is bound by a complementary region on the in the 16S component of the 30S ribosome. The first AUG codon downstream from the Shine-Dalgarno sequence is then recognized by the tRNA carrying F-met. Initiation also involves various proteins, the 50S ribosomal subunit, and it requires GTP hydrolysis. So what we're looking at here is a figure that describes or shows us visually the complex process of ribosomal assembly that must occur prior to the initiation of protein synthesis. Here's the 30S ribosome. And in prokaryotes, mRNA, the first, or the FMET, tRNA, GTP, and a 30S ribosomal subunit all come together with the assistance of 3 initiation factors. We have initiation factor 1, 2, and 3. And this helps, or this does form that 30S initiation complex. So the 30S subunit binds specifically to the Shindell-Garneau sequence, and that is shown in red. Well, it's not really red, it's kind of orange in my mind. But near the 5' end of the mRNA transcript. And what that does is orients the FMET tRNA on the first AUG codon, which is shown in green. So here's that green. And I failed to mention, once that the 30S is formed to this initiation complex, IF3 is released. Once the tRNA and FMET are positioned over the Shindell-Garneau sequence and the AUG, then the ribosome, the 50S ribosomal subunit comes in. The initiation factors IF1 and IF2, and these are proteins, by the way, are released, and GTP is hydrolyzed to GDP, an inorganic phosphate. So this is what provides the energy for this process. Then the complete 70S ribosome is now assembled. The FMET tRNA is at the right place on the first AUG codon. So everything now is ready for that elongation process. And the last slide that we'll look at in this video, there will be one more. I think I'll only make it one. Oh, sorry. Forgot. Whoopsie. Whoops. Uh-oh. I can't go back. Well, I'm going to just talk then, and I'm sorry I don't have this up here. But there is a slide that shows the Shindell-Garneau sequence and initiation code word established in that mRNA reading frame. You can see the Shindell-Garneau sequence on the 5' end of the mRNA, and it's base pairing with a 7-base sequence, UCC-UCCA, near the 3' end of the 16S ribosomal RNA. Also, if you'll notice the proximity of that Shindell-Garneau sequence to the first AUG, and the importance of that Shindell-Garneau sequence is that it results in the initiation of translation at the nearest AUG. So this ensures synthesis proceeds from the correct codon and in the right reading frame. Sorry I flipped through those slides, I didn't mean to, but we'll just conclude here. Thanks.